Introduce our second keynote of the meeting, Bob Waterston. So Bob is one of the founders of the field of genome biology, and I think by extension, effectively one of the founders of this meeting. But it's nice to have the opportunity to be able to introduce him and tell you more about him than I'm sure many of you know. So Bob grew up in the suburbs of Detroit. Maybe before there were suburbs, I don't know. That's what he went to Princeton University, and there, as you'll see, is evident from his LeBron-like physique, played both basketball and football. After Princeton, he went to the University of Chicago for medical school, where he got distracted by biology and ended up completing his MD, but also doing a PhD, so an MD PhD before that was really a program. And then after that, I had some more history here, but I think he's going to tell you about it in some of his slides, so I'm gonna leave it out. But let's fast forward a little bit. Ended up working on worms, and ultimately at WashU as faculty, where he worked in a close partnership with Sidney Brenner. With John Sulston on the mapping and ultimately sequencing of the C.elegans genome, right? And of course, this project of C.elegans was the first animal multicellular organism genome to be completed, and effectively laid the groundwork both from a technological and executional point of view for the Human Genome Project. So in the midst of all this, I think Bob got distracted away from worm for a little while to get involved with the Human Genome Project. And for that critical period leading up to 2001, he led the WashU built and led the WashU Genome Sequencing Center, which together with the Sanger Center produced the majority of the finished sequence for the human genome. So I think one thing that kind of gets a little bit lost in the history here. Well, there's two things. One is I only learned about this really from reading a little bit about Bob, and despite knowing him for a long time. So during the Genome Project, he actually, during when it was accelerating and the competition with Celera was heating up, he actually had a life-threatening illness and was in the hospital for a good chunk of it. And he still took his conference calls from a mini office in the hospital every Friday. So the next time you're debating whether to call into your Uncode call, think of that. So, are you committed? So the other thing I'll point out is I think we often as a field take it for granted, the notion of open data sharing, of public release of data, of just generally being early and open about everything, is kind of an ethos that is still somewhat unique within science. And certainly did not always exist. And if you look back on the origins of that, a lot of it has to do with even before the Human Genome Project, the C.elegans Genome Project, where John and Bob were real leaders in championing the free and immediate release of data well before publication, and that in turn led the groundwork for the Bermuda Principles and that same policy through the Human Genome Project. And I think ultimately kind of the ethos that has caught on and stayed with us, and now I think is in a way propagating outward from genomics into the rest of the field. Somehow I didn't get to advance my slides here. So there's Bob and John sitting. I think that's in one of the early AGBT meetings, if I'm not mistaken. And then another shot here of this is at the White House, I think. Everyone's younger, but around the, I see you, Richard, around the time of the completion of the draft of the Human Genome Project. So, it's possible that and somewhere along, this entire decade and a half or so while he was leading the genome, or helping to lead the Genome Project, he was also chair at WashU and built a really amazing department there. But I think it's possible that the degree of friction and interactions during the Genome Project made him think about a move. There he is with with Eric talking at the same time. And, before I was there, but ultimately to what would be to my great benefit, he, in 2003, in the wake of the Human Genome Project and also the Mouse Genome Project, which he co-led, left one Washington for the other Washington, moving to Seattle, where he founded the Department of Genome Sciences and has been, until last year, the Bill Gates Endowed Chair of Genome Sciences. And so the reason he hasn't been coming to this meeting for the last decade has been because he's had to go to a chairs meeting the same week, right? So we're finally, he's finally stepped down, and we're glad to get him back here. So, on a more kind of so in terms of awards, Bob, too many to list, but I'll just mention a couple. So for the work with John Sulston and Sidney Brenner on the worm genome, he received the Dan David Prize. For work on contributions to that genome as well as the Human Genome Project, he, along with 7 other individuals, received the Gairdner Award in 2002. And then more recently, he received the Gruber Prize in genetics. One of the more even rarer distinctions that I like to point out, especially since I ran into this webpage randomly. He's one of the only scientists to be honored by appearing in The Onion, where they somehow misnamed him Louis Farland, but you can clearly see the genome there, and in typical Bob what the hell fashion. So I'll just, Bob's going to talk about, I think, the science that he's done in the years since the Genome Project. So I'm not going to talk too much about that, but I just want to say a few kind of personal words about him and sort of my experiences over the last decade of being at UW with him as my chair. So he was the one who hired me. I had, so I've gotten to know him very well, both through that as well as by teaching with him for, I think, 7 or 8 years, the introductory class for genome scientists to first-year graduate students. I was so intimidated by him the first couple years that I was there, and kind of the breadth of knowledge, that I was basically a silent student in the class, to the point that apparently the running joke was that it was Bob and Silent J. So, but I've learned. I think I learned more from that class than I did practically in graduate school, just from absorbing. And then as a chair and as a colleague, it's really difficult to put into words kind of what a spectacular leader and effectively mentor he's been to me and other faculty there, in addition to his own lab. I think I've learned more from him possibly than anyone, not partly science, but also how to be in science, right? And how to be a colleague, and how to treat your students, and all of that stuff. And the other thing I admire is just it's almost like he's a PI for the entire department, and he follows the work of 300 or 400 people in kind of intricate detail. So I don't know how he does it. So with that, I'll let Bob talk, and looking forward to hearing your keynote.
I should just go home. It's only downhill from there. Let's see. Oh, this way up. I'm on, right? But I have to log in. I have movies, and they don't work very well. All right. Well, first of all, thank you very much for the honor of addressing you. It is indeed a pleasure to be back here. It's been a great meeting. The science is terrific. It's been fun to watch how it's changed. But I noticed that ultra-conserved sequences were a topic when I last attended. Anyway, so I'm going to tell you today about our efforts trying to define the transcriptional program for the C.elegans embryo. But as the subtitle suggests, I'm going to tell you a little bit, give you a little perspective and motivation for the work that follows. So my life with worms starts with Sidney. Sidney was a fantastic scientist. And famously, in 1963, he wrote a proposal to the MRC proposing to switch from his enormously successful studies of bacteria and phage to study an animal. And he said that the animal, the organism, should have relatively few cells so that exhaustive studies of lineage and pattern can be made. And of course, the animal he was talking about was eventually C. elegans. And his choice has been excellent. And I should say that I first learned of the worm as a student at Woods Hole in the physiology course. Sidney was there giving a class every day talking about the literature. He would talk about whatever we had asked him to talk about the day before. He was incredibly impressive with his knowledge of whatever we asked him to talk about. We could have asked him to talk about a movie, and I think it would have been equally entertaining. But anyway, at the end of the summer, he gave a lecture, first public lecture on his work on C. elegans in Woods Hole that summer, and I was captivated. I went up to him finally at the end of the summer and asked him if I could come and do a postdoc with him. And he just sort of looked bemused and said, "Write to me when you get home." And I thought, oh, shit. But I wrote, and he took me. So I started with worms. And of course, worms are famous for, as he thought about, they're famous for having complete and comprehensive data sets. And one of the most famous is indeed the lineage. And John Sulston sat in a dark room for 2 years, watching worms like this, taking notes of their divisions. These are time points a few minutes apart. And he used that to generate the lineage of all 558 cells of the worm. This is just a portion of it. The whole thing is you can't see on this scale. And that's been a fantastic resource. And then also, the worm has a whole series of complicated behaviors. John White and Nickel Thompson, along with Eileen Southgate, did EM sections of the worm. I think the longest series they did was 6,000 EM sections. I don't know if any of you have ever done EM, but Nickel did not lose a single one. Anyway, they would trace the outlines of the neurons in every section and then move on to the next and thereby got the connectivity of the worm, all 302 neurons. Not very many, but it works. Another one is the C. elegans genome, and I mention that because this is how I got involved. John Sulston, having finished the worm, decided that he needed to do a physical map of the worm. And his motivation was that because of the powerful genetics, people were finding lots of mutants, but the hang-up was trying to get the DNA, trying to understand really what was going on, cloning it. And he really reasoned that if he had a physical map of the whole genome, people could just look up what was going on. And so he took genomic libraries of the worm, digested them with enzymes to produce patterns like this. The details aren't important, but they could detect overlaps between clones and with those overlaps build up the genome. But by the time I got there on sabbatical in 1985, it was obvious that there was a problem. And that problem was that certain regions of the worm genome just didn't propagate in E. coli. So that after processing 17,500 clones, and this is one at a time sort of thing, not a small number, theoretically, it should have had 99% coverage and been very close to complete. Instead, it only covered 80% of the genome or something like that, and they had over 700 gaps in the sequence or in the map. And so when I was there, I spent time variety of approaches trying to figure out how we might solve this, and without much success. But by the end of the summer, by the end of my year there, Maynard Olson and his graduate student David Burke had developed a yeast artificial chromosome cloning system. And the pieces were not only bigger than they were in E. coli, it was a eukaryotic host. And certainly the biases would be different. And that proved to be true. I got home and made a YAC library, started collaborating with John and Alan, sent them clones. It was the beginning of a really marvelous decade and a half collaboration. And the YACs closed all but 5 gaps. And so in 1989, this is the back of that hall down there, Bush. Alan Coulson, who was working with John and had formerly worked with Fred Sanger, did all that. Alan printed out all these contigs on A4 paper, sellotaped them together, high tech. And this is the first chromosome, second chromosome, the X chromosome. And you can see the continuity is pretty good. 2 goes from one end of the hall to the other. We have a hole here, but it's probably down there. Anyway, it was great because everybody walked up to the map and tried to find their favorite little clone and wrote things down, and it was fun. But we had another motivation for doing this, and that was earlier in the year, Jim Watson had been talking to people and let it be known that he was going to include yeast, flies, and E. coli along with humans in the Human Genome Project. But he didn't think worms should be part of it. So this is bad news for the worm. So we got together and decided that if we could show Jim a map like this, maybe we could change his mind. And we made sure he came and saw it. And reportedly, I wasn't there, but I have it on good authority. He said, you can't see it without wanting to sequence it, can you? So we knew, and we talked to him on Saturday, and he said he would entertain a proposal from us. And so that started things off. And 8 years later, as Jay alluded to, by improving methods, by increasing our group sizes, we doubled our sequence output every year, basically. And we had the genome sequenced by 98. One of the surprising things was that the worm had as many genes as the human, not the 4,000 that genetics had estimated. And as Jay said, another gratifying thing was that the methods that we had developed for the worm were applicable to the human. And we went on and participated in that, as he alluded to. But the worm, I think, gets credit for another thing, and Jay alluded to this a little bit, but I want to emphasize where it all comes from. And that is that the worm community had a marvelous tradition of sharing things, pre-publication. And this is the Worm Breeders Gazette. My mother could never understand what it was about. This says Casino Rhabditis. It had covers like this. There was various movies and so forth. It was a collection of abstracts that came out every 6 months, and it had ideas and results that everybody shared. And it was in that spirit of sharing that John started sharing all the map data as it came out. And when we started sequencing, we kept the same policy. And with the advances in the internet, we could now share things almost daily. And fortunately, as Jay alluded to, that became the policy of the Human Genome Project at the series of Bermuda meetings. And there's recently been a nice, extensive article on all this by Kate, Max, and Jones, describing really how open data came to be. All right, so When I moved to Seattle, I left genomes behind me, at least genome sequencing, and started focusing on trying to figure out what the genome of the worm actually did. We had all these letters, we couldn't read it. So I've been doing a variety of things. I'm going to focus today on our efforts on the transcriptome. One of the things we did was early on, we did an extensive time course, so we could find out at least when the genes were expressed. More recently, we used fluorescently labeled cells and FACS sorting to recover specific cell types and to watch them over time in the embryo. But what I'm going to spend most of the time on is in single cell analysis of expression in the worm. And we've been doing this for a long time, well before single cell sequencing. And this is, I hope I can do this without, wait a minute. There we go. Oh. It's not going to let me do this. Is it going to? How can I do this? Oh, there it goes. No, space will take me to. Yeah, space might do it. Anyway, so, nope, that's what happens. All right, we'll try it again. No. Damn. There we go. These are nuclei labeled with histone GFP. And so you can watch what John had to look down with Namarsky optics. We have this high contrast image, and we can collect these and analyze them with a program Jurong Bao developed. And if I dare stop this, no. Oh, I know, I can just put it out here. Yeah. And into the same background, we've introduced a second construct with a different color. And this is being driven by promoters from the gene DMD4. It's a transcription factor. And it's coming on in these cells and nowhere else. Okay? And I don't, there's probably, it comes on and so forth. We can take all this image data and reduce it to this. So this is the lineage of that animal reconstructed from that image. And this is when that gene is first detected, and these are the cells. And from the lineage, we know what the cells are. And so we know that this is going to become posterior cells in the pharynx, and we know what kind. So we've done this on something like 280 different transcription factors. But it is hard work. It's beautiful data, but it's really slow. So we've been interested in the idea of single-cell sequencing for a long time. Because obviously, if we can do this on all the genes in single cells, we could reconstruct this. We could have a lineage like this, not just for the 281 genes, but for all of them. So single-cell sequencing in elegans, we learned early on that worm cells are very small. The cells in the embryo have probably on the order of 20,000 mRNA molecules, about a tenth that or 25th of that of mammalian cells, certainly cultured cells. And with most RNA single-cell sequencing methods, you sample only a small fraction. So we recover only a very few UMIs per cell. And the other thing is that when you dissociate the cells in the embryo, you lose track of them. You can keep track of them at the 2-cell stage and the 4-cell stage, and if you're really heroic, you can probably do it at the 8-cell stage. I can't. And we want to do the whole thing, and so we lose track. So we knew for all this to work and to deal with the complexity of the whole animal, we needed many copies of the same cell type to obtain an accurate profile, and we needed good expression markers. In order to identify cells, and we'd have to do it by RNA alone. You're familiar with that idea. So when Jay's student, Jun Kao, came down the hall saying that he had a method that would do tens of thousands of cells, we said, we have the system you should use. And fortunately, we convinced them that this was a good thing. And so a couple of years ago, we published the single-cell transcriptional profiling of the second larval stage. And we picked that because the cells are stable, they're not changing, and they're terminally differentiated. And the cells are bigger than they are in the embryo, and the gonad hasn't started to develop. So we didn't have to compete with all those. And so by using markers and clustering methods and so forth, we could indeed identify the major tissues of the worm. And if we took the neuron cluster or any of these other clusters and subclustered them, we got more detailed information. And here I'm showing you the neuron cluster and highlighting this 1 and 2 cluster, which are cells that are represented once in the embryo. So we have the sensitivity to see single cells, and we can identify them. These are 2 neurons. They're left and right neurons in the worm. And people knew that they were different, and we could find those differences and more between them. So that was exciting. And I can't remember how many cell types altogether we could discover. We've gone back and analyzed the data. New methods are even better. But we wanted now to take on the harder task of seeing if we could profile the embryo. And remember, it's dynamic. The cells get very small. And we knew this was going to be a challenge. And we tried various methods. We tried the combinatoric indexing method, and we just couldn't get enough UMIs to get accurate identification. And so we had some success with the 10x instrument, and we returned to that. And so this is how we do it, basically. So we synchronize the growth of worms. We get adults just beginning to lay eggs. We take those adults, and we don't really cut them open like that, but it's cute. Anyway, we get the eggs out. We let the eggs develop for a desired period of time, because they're all more or less early pre-gastrulation embryos. And then we digest the eggshell and dissociate all the cells. And we do this from not one animal, but thousands or even hundreds of thousands of worms to get cells. We then load those on the 10x, and we make libraries in sequence, things you know about. All right, and this is the work. I should point out that this work has been done by Jonathan Packer and Xiao Hun. Xiao's done the wet lab stuff. Jonathan is amazing at doing the computing, and he knows more about worm embryogenesis and markers for it than I could hope for. Anyway, so we set out and we collected 4 different samples. We wanted to be sure we covered this kind of area deeply. And just when we were starting to get deep into the analysis of the data, I heard from my former postdoc, John Murray, that he had been doing things, and they had collected a broad time series and had 3 data sets, and they were working with Junyoung and Chin. And so we said we'd throw all our data together and see what we could find. It's been great. So we've got 84,000 cells after all the QC and throwing away doublets and all that kind of stuff, which represents 60-fold oversampling of all the cells in the embryo, not just the terminal cells, but all their ancestors. And that's been key. So this is fun. I hope this will work. These are basically the dispersed cells before any analysis. We've colored them by the age of the cell, but the program doesn't know that. And we've aged them simply by comparison to our early embryo time series. Each cell we do a correlation. And Cole Trapnell did this lovely movie for us. There we go. So the cells are magically trying to cluster together. This is obviously not how it works, but, but it's pretty cool. And so we get this amazingly fine grain structure. And remarkably, we go from green to red to, green to blue to red on each of these strands. There we go. And so we get a temporal trajectory on all of these things. So we're following the development. We have enough density and we have enough sampling to just follow the whole thing. And if we, whoops, I need to. There, oh. Let's try that. Anyway, this is again just fun. You drive around through this, you can see these are muscle cells, these are anterior muscle cells, these are ciliated neurons taking off, that's germline, and so forth. It's fun to look at it like that. All right, enough of that. Back to 2D. It's a little easier to look at things and analyze. We can use markers and identify all these different major cell types. No problem. But what we wanted to do was see if we could do more. And so we subclustered again. And we're going to take the ciliated neurons here. And we're select those, and we're going to subcluster them. And from that, we get this picture. And each of these is a lovingly named neuron. They all have these three-letter names. And they're mostly bilaterally symmetric. Some of them are four-fold symmetric. Some of them are six-fold symmetric. And some of them are unique, the ASEL and ASER. So Jonathan, there are only 22 known morphological, based on morphology, there are 22 ciliated neurons. We can identify 21. We can't get the PHA, but we think we might have it. Okay. And so, these are the ADL neurons are represented twice, the ASG twice. So we have really good resolution. But you'll notice, and altogether, doing this repeatedly on different tissues, different subclusters, we can identify 105 distinct cell types. But you'll notice here that there are almost lineage-like relationships, and that turns out to be true. And so this is the parent of those two cells. This is the grandparent, and so forth. We wanted to see if we could do this now across the whole of development. And while Jonathan has focused on identifying all these terminal cells, John Murray focused on identifying the lineages. And so we took all these early cells, primarily from the AB lineage, less than 250 minutes for those who know worms, and we got a picture like this. And these are different progeny of the ABA neuron. I probably should give you a brief tutorial on worms, worm lineages. So it goes from 1 to 2 to 4 to 8, with AB dividing anterior, posterior, and then left, right. And that's represented obviously like this. But the naming is such that this is the left daughter of the anterior daughter of AB. So the numbers of letters correspond to the number of generations, and it gives you the position. Okay, so now you can see that this is one progenitor. So this is when AB has 8 cells. This is a symmetric lineage, so the left and right lineages give rise to the same kinds of cells, and we can't distinguish. We don't see any difference between left and right. And the same here. Here they're asymmetric, and we can see the differences. Okay, so John heroically went through this and identified all these cells. I hope you memorized this. I know you can't even see it, but it's all the cells of the ABPXP lineage and several of the generations. So let's take a look at just this bit here so you can see it better. I blew it up. And here are the marker genes that are used. You can see their expression patterns. And here you can see the lineages they correspond to. So the RNA is enough, and the software is good enough to reconstruct the lineage from the sequence. It's quite remarkable. I should say that it doesn't always work this way, that we get fakes, and we have to give it the right set of cells to get the right answer sometimes. But we know what the answer should be. So at this point, we have 105 terminal cell types identified. 70% of neurons, 68 out of 96. We're missing the pharyngeal neurons. We see their precursors, but we don't see the mature pharyngeal neurons. And we think that's because they become buried in other pharyngeal cells. And we're going back and looking at the doublets that we threw out to see if we can identify those, because we now have their precursors and we know what genes to look for. With John's efforts, we have basically all the pre-terminal lineages and the cells contained in them from about the 50-cell stage to terminal differentiation. And these two things were done independently, but the lineage gives rise to the right cells and connects to the right cells. So it gives us great confidence that the answer we're getting is actually right. Okay, so this is a great resource. We hope everybody uses it. We hope people use it to develop even better algorithms than UMAP, things that will do things automatically that we've had to do by hand in figuring out the lineage. But there have been some interesting biological things coming out of it too. And one is that cell lineages have a gene expression signature. One lineage is distinct from another, but that's lost as the cells undergo their final terminal differentiation. And we also get, these are terminal cells down here, the IL-1 and IL-2 neurons. They come from 3 different lineages. And so the lineages are quite distinct to begin with. They converge on the final common cell type. And we think that's probably a feature of many different organisms. But I want to spend time on the last one here in a little more detail. We see lineages that appear to differentiate very quickly. So instead of having a smooth trajectory, we see things disrupted. And we think this may be facilitated by multi-lineage priming, which we see, and I'll show you that. Okay, so here, these are the parents of the IL socket cells. They're glial cells. And these are the ventral ones. These are parents of the dorsal ones. So they don't have this nice connection that we saw in the ciliated neurons. And so what's happening there? Here's another example. These are the parents, and these are the progeny. They don't form nice patterns like this one. But let's go back to this because we do have this and we can see what's going on. So let's look at that. This is really this lineage. So these are the grandparents. This is the parent of these 2 cells, and this is parent and so forth down here. And so we're going to look at the expression of transcription factors as we traverse those lineages. And so, you know, we really do have expression profiles of every gene for every lineage at this point. And so you see here, I should explain a little bit what we're doing here. So this is the grandparent, and then you see the parent cells, and then the daughter cells. And you can see by the color, it's hard to see, but that's, anyway, I've colored the lineage accordingly. So to begin with, you see multiple transcription factors that differ between the lineages. These are not unique in the worm. They are not specific for these lineage, but they're specific for the sister-cousin set, right? So, and it's not one, it's multiple transcription factors that are being involved. But if you look at the bottom, we see transcription factors that are on in the parent and in one of the daughters, but not the other. And then here's one going the other way. And here's one that's in this and this, but then lost there. Okay, so this is what we're thinking of as multi-lineage priming, that the gene is on here, but in that context, it doesn't drive differentiation. And it's only when it becomes in this final division and gets isolated from the other things that it actually leads to differentiation or participates in it. And just to show you what it looks like on the data, on that map, we've colored things according to whether they're specific to one or the other, or whether they have both. And you can see this lineage has just the blue, and this has the red, and this has blue. And again, you can see they're not specific overall, but they're only specific within the lineage. Okay, and one other point about this is that we wonder if it's, first, can we confirm all this? Is this really real? And one of our movies actually shows this for HLH3. It goes down, it's in this branch, and goes off in this branch. It's not that we don't think that this is necessarily segregation of the message. We think this could be accounted for simply by degradation, normal degradation, and continued expression. There could be segregation, but we don't have any evidence of it. So, is this weird, or is this a one-off? Well, actually, it's fairly pervasive. About 40% in the AB generation 7 of those TFs, about 40% of those are selectively retained in one daughter and not the other in this. So that's my story. Just to summarize, we've done the RNA-seq of more than 80,000 cells spanning from mid-gastrulation to terminal differentiation. We can identify more than 105 different cell types. The number keeps going up. We have gene expression profiles throughout, and we know what's in each cell. We know what transcription factors are on. We know what other targets are. And the dynamics of expression show this loss of lineage signature, convergence of different lineages, and this multi-lineage priming. So, and with that, I'd just like to thank people again. Chow and Jonathan have been fantastic. It's been a great collaboration with John Murray and colleagues at Penn. Funding sources, NIGMS, NHGRI, I should have put, NHGRI and Bill Gates. And then I would like to also just acknowledge my great debt to John Sulston. He died last year, about a couple months earlier than this time of year. I owe so much to him. He was a fantastic scientist and a great friend, and I miss him. Thank you.
Questions for Bob?
Questions?
So first of all, beautiful work, and it's really nice that sort of you can begin to see some principles, for example, the multi-lineage priming. And my question is that given how different C.elegans is from, let's say, the mammals in terms of its regulatory structure of its promoters, in terms of the determinism of cell fate choice, how much do you think of what we learned here in terms of the principles and how these transcriptional decisions work in an animal whose sort of both sequence logic and actual deterministic behavior is so different? And what do you sort of envision sort of translating to other animals such as the mammals?
I mean, clearly C. elegans is pared down things. And I think that the reason they have this fixed lineage, not all nematodes do, is that they need to do things in such a hurry. This whole development takes place in 12 hours. Those cell divisions last 30 minutes. And think about how many messages you have to make in that time. It's a little mind-boggling, and you only have 20, anyway. So yeah, there are going to be specialized things, but something like the multi-lineage priming, that's been observed elsewhere.
So as the convergence.
And so I was going to say, we put our paper up on bioRxiv, and 2 days later somebody said we saw convergence too, and they were very excited about it. And so I think you'll have to see. But I think, you know, the idea that it's multiple transcription factors that are doing the trick, these are not surprising. It's nice to see it, though.
Questions?
I have a question of my own. Thanks for the great talk. Do you think that you could shed some light on the previous topic by tracking somatic mutations with single-cell sequencing along that entire beautiful lineage?
Boy, I suppose. But, I mean, the time frame is just so different. Molly was talking about cell division every 2 days. The worm is done in 3 days. The worm goes from egg to egg-laying adult in 3 days. I don't know how much somatic mutation there should be. Some people are beginning, there are people actually trying to look at that kind of thing by introducing mutation-sensitive reporters. So you can actually see something happen.
If I may ask here, about 10% of the cells die by apoptosis. One of them was the ASC sister cell.
Yeah.
Do you see any, a cluster, for example, of apoptotic cells, or?
Now, we've looked hard for cell death, and in the HMC cell, its sister, its contralateral cell dies, and so we can see it beginning to form, and then it just disappears, and we don't, even there we don't see a signature. We think that, I mean, the cell death in worms is very fast.
Very fast, but perhaps in a SED3 background, or if you keep them around.
That's an experiment we're thinking about, is to put it in a SED3 background and answer the question, what actually goes into the dead cells. Yeah. SED3 is a mutation that knocks out, that prevents cells from dying.
So,
yes, Richard. Uh-oh, I don't know what Richard's going to ask. He knows too much.
No, I'm really struck by how continuous, you know, so you said that there's some that jump, but how there's this trajectory through the lifetime of a cell.
And its grandparents. And
yeah, so that the state, it isn't a sort of discrete state switch from cell to cell, but it's priming itself. It's on a track the whole way to get somewhere. And
we don't see very many disruptions. The jumps that I talked about are the exception, fortunately.
And so you can really position things very precisely in time, probably, in that state.
Yes, and our time estimates actually match up remarkably well with the cell divisions. So we see, you know, like that AUA, ASG division. Our estimates of those cell times actually match up with John's lineage.
What was I going to ask?
Following up your point about how fast worms live, so a lot of the headlines we see about like transgenerational inheritance they end up being from worms, where 14 generations is short and might actually mean something in like seasonal adaptation terms.
Yeah.
So have you started to think about how you might look at how that question further complicates the trajectories you see if you can you do this in parallel with kind of epigenetically primed populations?
Yeah, we hadn't thought about that, but that's an interesting idea. Yeah, I think you could do it. Yeah, and see what changed. Yeah, I mean, you know, we don't have hundreds of cells, and we don't have thousands of. I mean, we don't have hundreds of thousands of counts per gene. So our estimates can have
noise already.
error bars on them, bigger than we want sometimes. But if the gene is reasonably expressed, we can nail it.
Okay, Bob, I want to ask about intercellular interaction, let's say receptor-ligand crosstalk, whether that can partly explain the abrupt change of lineage character, so the cell suddenly forgets its ancestry and do what the neighbor tells them to do.
So there's crosstalk all along at those AB anterior-posterior divisions. That's Wnt signaling that is distinguishing the anterior from posterior. And there we see quite, through those divisions, as Richard was pointing out, we see quite continuous things. It's only at the very terminal division that we see this disruption. And when we look, that cell has a lot of differential gene products already.
So could the cell ablation experiment help to sort that out?
Yeah, possibly, yeah.
Two more questions from there, and then.
Okay, so in the human single-cell sequencing world, I feel like there are a lot of questions about what is a cell type or what is just a different cell state. Do you think that we can use a map like this where we know what the cell types are to think about how much variance we expect between different cell types versus what would just be a different cell state of the same cell?
We hope so. and we hope also that people will try out algorithms for mapping and detecting these relationships. We see both false and false positive. We see things that look like they're dividing, and we know they're not. And so we hope that this kind of data will provide a ground truth that will stimulate things on human and mouse.
I'm really curious about these cases where different cell types, which you know by lineage, are separated, but by RNA they kind of go together.
They converge.
Yeah, they converge. Well, not only that they sort of terminally converge, but are there cases which during the lineage they converge, but then split again? So in other words, where the RNA state is not adequate to describe the sort of cellular trajectory, and I guess there's going to be some statistical noise, so I guess it's going to be hard to find such cases. But are there cases which kind of go together and then split apart, implying?
Yes. So I didn't go into it, but on that glial lineage, there's actually a cluster of cells at the base, and that cluster we think comes from different lineages, but then gives rise to 3 different kinds of glial cells. So we believe that would be a precursor state to a common precursor state for glial cells. And then the later program is superimposed on that. Does that make sense? It does. And we see that in a couple other places too.
Okay, let's thank Bob and Molly again for 2 wonderful keynotes.